40% of women in their 40s have microscopic breast cancers inside their bodies right now. Not a risk, not a possibility, a documented biological reality, confirmed in autopsies of women who died from unrelated causes, with no diagnosis, no symptoms, no awareness. Most of those cancers never became dangerous, not because they were not there, because 3 internal defense systems kept them controlled. After menopause, 2 of those 3 systems lose efficiency at the same time. The reason is the same for both. Estrogen was suppressing chronic inflammation and sustaining immune cell activity, work it had been doing silently for decades, work that most appointments have never had time to describe. Without it, the internal environment shifts. The fire that was burning low finds less resistance. The immune guards that were eliminating abnormal cells around the clock begin working shorter shifts. And the microscopic cancers that were dormant receive an environment that is, for the first time, favorable to growth. It is why women with no family history of cancer are diagnosed after 60 at rates that do not match their perceived risk. It is why doing everything right on the outside, eating carefully, staying active, attending every screening, does not change what is happening inside, the internal environment that menopause altered. And it is why that gap between what women are told to do and what the biology actually requires carries a weight that feels, quietly, like something is being missed. Because something is. I am Dr. Elizabeth Carter. I am a board-certified internal medicine physician with a fellowship in women's health and midlife medicine and 32 years of clinical experience treating women at exactly this stage of life. What the peer-reviewed research from Harvard, Johns Hopkins, UCLA, Ohio State, and Brigham Young University identifies as the specific dietary response to all 3 of these defense systems simultaneously is not something a 15-minute appointment has time to cover. That is why this channel exists. In this video, I am going to show you 5 foods, the exact amounts the studies used, and the one preparation mistake that eliminates the benefit of 2 of them before the first bite. If this kind of evidence-based analysis is useful to you, please subscribe. I post 2 to 3 times a week. 2 seconds. Do it now. Then stay with me, because what comes next is the explanation I wish every woman over 60 had received the year her estrogen fell. Microscopic cancers are forming inside your body right now. Not might be forming, are forming. This is established science, documented in autopsies of people who died from accidents with no cancer diagnosis, no symptoms, no awareness. 40% of women in their 40s have microscopic breast cancers. 70% of people in their 70s have microscopic thyroid cancers. Most of them never become clinically dangerous, not because they were not there, because 3 defense systems kept them controlled. After menopause, 2 of those 3 systems lose efficiency simultaneously, and the reason is the same for both. Estrogen was doing work inside your immune system and your inflammatory response that most appointments have never had time to explain. Without it, the internal environment changes. What was controlled may not stay controlled. The biology does not announce this. It simply shifts. The process is called chronic inflammatory overload. It is the environment that microscopic cancers need to grow. Estrogen was suppressing that environment for decades. After menopause, that suppression weakens. The fire that was always burning low finds less resistance. Genetics defines the risk. The internal environment defines the outcome. And the internal environment can be changed. 5 specific foods, in specific amounts, prepared correctly, activate all 3 defense systems simultaneously. The research on each one is published in peer-reviewed journals from institutions including Harvard, Johns Hopkins, UCLA, Ohio State, and Brigham Young University. The doses are not estimates. They are the exact amounts that produced measurable results in real people, in real studies. Here are the three systems your body has been using to keep microscopic cancers dormant. And here is what menopause did to two of them. The first system is your immune surveillance. Natural killer cells, T cells, and macrophages patrol your body continuously, identifying and eliminating abnormal cells before they can grow. When they find a microscopic cancer, they eliminate it. This happens thousands of times during the lifetime without any symptom. Think of them as security guards checking every room in a large building, around the clock. The moment they detect an intruder, they remove it. After menopause, natural killer cell activity declines. Estrogen had a direct role in maintaining immune function that is only now being documented in clinical literature. The guards are still there, but there are fewer of them, and they are working shorter shifts. The second system is inflammation control. Acute inflammation is protective. When you cut your finger, your immune system floods the area to prevent infection and begin healing. Chronic inflammation is different. It is a small fire that never fully extinguishes. That fire releases chemical signals that tell cells to grow faster, divide more rapidly, and ignore the normal stop signals that healthy tissue obeys. Microscopic cancers respond to that environment the way a fire responds to gasoline. They do not create the fire, they require it to expand. Estrogen was the suppressor that kept that fire low. After menopause, the suppressor is gone. The fire burns with less resistance. The third system is anti-angiogenesis. This is the one most consultations never have time to explain. Microscopic cancers cannot grow beyond approximately 2 millimeters, the size of a ballpoint pen tip, without blood vessels. Without a blood supply, they hit a size limit. They stay harmless indefinitely. But if a cancer can signal the body to grow blood vessels toward it, everything changes. Once blood vessels reach a tumor and begin feeding it oxygen and nutrients, that tumor can grow 16,000 times larger in as little as 2 weeks. 16,000 times. That is the difference between a harmless cluster of cells and a clinical diagnosis. The body has the capacity to refuse that signal. When anti-angiogenic defenses are strong, the blood vessels never grow. When they are weakened, they grow, and the cancer expands. Each of the 5 foods I am going to show you activates at least 2 of these 3 systems. Some activate all 3. The first one does. Green tea contains a compound called EGCG, epigallocatechin gallate. It is a natural molecule found in tea leaves that dissolves into the water when you brew correctly. And I want to be precise here, because this is where most people lose the majority of the benefit before the first sip. Never pour boiling water over tea leaves or tea bags. Boiling water damages the catechins. The correct temperature is approximately 175 degrees Fahrenheit. If you do not have a thermometer, bring water to a boil, then let it sit for 3 minutes before pouring. Steep for 5 minutes. If you are using tea bags, dunk them repeatedly during the steep. You are physically releasing the catechins from the leaves. Now here is what EGCG does when prepared correctly. Researchers at the University of California measured inflammatory markers in people before and after drinking green tea daily for 8 weeks. They found reductions of 30 to 50% in C-reactive protein, interleukin-6, and tumor necrosis factor alpha. Those are the 3 primary systemic inflammatory markers. 30 to 50% reduction from a beverage in 8 weeks. The fire came down measurably. A study at Brigham Young University gave older adults 3 cups of green tea daily for 3 months. At the end, they measured natural killer cell function. It had increased by 47%. The guards who had been working shorter shifts came back to full capacity. Nearly half again as effective at identifying and eliminating abnormal cells. And in population studies in Japan, tracking over 8,000 people for 10 years, those drinking 5 or more cups of green tea daily had 40% lower rates of colon cancer, 30% lower rates of gastric cancer, and 50% lower rates of esophageal cancer compared to those drinking less than 1 cup daily. The protective effect was concentrated in digestive cancers because green tea flows through the entire digestive tract. Every surface it contacts receives direct exposure to the catechins. It is, in the most literal clinical sense, coating the digestive system with protection. The therapeutic dose from these studies is 2 to 3 cups daily. For maximum effect, consider matcha powder. Matcha is ground whole tea leaf. 1 cup of matcha delivers the catechin content of approximately 10 cups of regular brewed green tea. The effective dose is 1 teaspoon of matcha whisked into hot water once daily. The second food is something most women already buy every week, and most are discarding the part that contains twice the concentration of the active compound. Broccoli belongs to the cruciferous vegetable family, alongside cauliflower, Brussels sprouts, kale, and cabbage. What makes this family biologically significant is a group of sulfur-containing molecules called sulforaphane. Sulforaphane activates a genetic pathway called NRF2. NRF2 is the switch that instructs your cells to produce their own internal antioxidants, not antioxidants consumed from outside. Antioxidants manufactured inside each cell, on demand, in response to a specific signal. Think of it this way. External antioxidant supplements arrive at the door and wait to be useful. Sulforaphane walks into the control room and turns on the internal factory. The antioxidants produced by NRF2 activation are orders of magnitude more targeted than anything that arrives from outside. Sulforaphane also increases natural killer cell production and activity, and it is anti-angiogenic. It prevents tumors from recruiting the blood vessels they need to expand. 3 systems, 1 compound. Now here's the clinical correction most women need. The florets of broccoli, the tree-like tops that most people eat, contain sulforaphane. The stalk contains twice as much. Most women cut off the florets and discard the stalk, or leave it on the plate. The stalk is where the concentrated medicine is. Slice it diagonally, saute it with garlic and olive oil, and the diagonal cut creates more surface area, which means it cooks faster and becomes tender. Or dice it and blend it into soup. The stalk should not be optional. It is the reason you bought the broccoli. Broccoli sprouts take this further. Sprouts are 3-day-old broccoli plants grown from seeds. They contain 100 times more sulforaphane than mature broccoli. The plant is born with all the sulforaphane it will ever have. As it grows, that sulforaphane distributes throughout an increasingly large structure. The sprout is concentrated. A study at Johns Hopkins University gave people with elevated cancer risk approximately 2 ounces of broccoli sprouts daily for 3 months. Cancer biomarkers in blood and urine dropped by 60%. Researchers also found that the sprouts had upregulated more than 200 cancer-fighting genes. 200 genes activated by a food. The effective dose from the studies is 1 cup of chopped broccoli daily, including the stalk, or half a cup of sprouts. The third food surprised the researchers who studied it, because most people think of it as a summer dessert rather than a clinical intervention. Papaya contains beta-cryptoxanthin, a carotenoid that preferentially accumulates in lung tissue. When you eat papaya regularly, the beta-cryptoxanthin enters the bloodstream and concentrates in the lungs, where it reduces local inflammation and inhibits the blood vessel growth that lung tumors require. Researchers at Harvard followed more than 70,000 women for 18 years. They tracked diet and cancer incidence. Women in the highest beta-cryptoxanthin intake group had 31% lower lung cancer risk than women in the lowest group. 31%. Over 18 years in 70,000 women. And the detail that changes the conversation, this effect was independent of smoking history. Women who had smoked earlier in life and increased their beta-cryptoxanthin intake still demonstrated the protective association. Beta-cryptoxanthin does not erase prior exposure, but it changes the internal environment that determines what happens to cells that were exposed. A word on fruit sugar, because this concern comes up frequently. Yes, cancer cells use glucose for energy. So do all cells, including the brain and muscles. The issue is not glucose itself. It is excess insulin produced by refined sugar and processed carbohydrates. Whole fruit contains fiber that slows glucose absorption. The insulin spike does not happen. Studies comparing women who eat whole fruit daily with women who avoid it consistently show that fruit eaters have lower cancer rates, not higher. Eat the papaya. Avoid fruit juice, which removes the fiber and concentrates the sugar. Whole fruit is protective. The effective dose from the studies is 1 cup of fresh papaya or 1.5 cups of frozen papaya daily. Before I continue to the fourth and fifth foods, the exact product I reference in this protocol is in the pinned comment directly below this video. It is the first thing you will see when you scroll down. Do not look for it anywhere else. The fourth food has a color that most people associate with novelty. The researchers at Ohio State University found something inside that color that explains one of the most important and least discussed questions in oncology, which is why cancers returned after treatment appears to have eliminated them. Purple potatoes contain anthocyanins, the same compounds that give blueberries their blue and red cabbage its red. Anthocyanins are anti-inflammatory and anti-angiogenic, but they have a third mechanism the other foods do not. Cancer stem cells are a small population of cells within a tumor that can regenerate the entire tumor. When someone receives chemotherapy, the treatment eliminates the bulk of cancer cells. The tumor shrinks or disappears. The person is declared free of disease. But if the cancer stem cells survive, the tumor can grow back. They are the roots of the dandelion. You can cut the dandelion at the surface. The roots remain. Anthocyanins from purple potatoes have been shown to eliminate cancer stem cells in colon cancer while leaving surrounding healthy cells unharmed. Researchers at Ohio State University exposed colon cancer stem cells to purple potato extract. The extract eliminated the stem cells. Normal colon cells were spared. Selective toxicity. In animal studies, tumors in animals receiving purple potato extract grew 65% more slowly than in animals receiving regular white potato extract. One additional benefit from preparation after cooking purple potatoes allowed them to cool completely and refrigerate before eating. Cooling converts some of the starch into resistant starch. Resistant starch causes a smaller insulin response and feeds beneficial gut bacteria, which ferment it and produce short-chain fatty acids that reduce systemic inflammation. The anti-cancer anthocyanins, plus resistant starch feeding inflammation-reducing gut bacteria. It is a cascade of effects from a single food, prepared one specific way. The effective dose is 1 medium purple potato or 1 cup of diced purple potato several times per week. The fifth food has been used medicinally for thousands of years. The research published in the last decade explains exactly why, at the cellular and microbial level. Pomegranate contains a class of compounds called elagitannins. When you eat pomegranate or drink the juice, the elagitannins travel to the colon, where gut bacteria convert them into smaller compounds called urolithins. The urolithins are what produce the biological effects. Researchers at UCLA gave pomegranate juice to men with prostate cancer who had been treated, but whose PSA levels were rising. Rising PSA after treatment is a signal that the cancer is returning. Before starting pomegranate juice, the average PSA doubling time in these men was 15 months. After drinking 8 ounces of pomegranate juice daily, the doubling time extended to 54 months. The cancer was still present, but its growth had slowed dramatically. Some men maintained stable PSA levels for years on pomegranate juice alone, avoiding the need for more aggressive intervention. The researchers identified the mechanism as anti-angiogenic. The allegitannins were starving the microscopic cancer cells by preventing blood vessel recruitment. The tumor could not expand because the supply line never arrived. But there is a second mechanism that is, in clinical terms, even more significant. The allegitannins in pomegranate feed a specific gut bacterium called Akkermansia muciniphila. Akkermansia lives in the mucus layer of the intestinal wall. It communicates with the immune system through that wall, sending signals that activate cancer-fighting immune cells. Studies in cancer patients receiving immunotherapy, treatments that direct the immune system to attack cancer, found that patients who respond well all have high levels of Akkermansia. Patients who do not respond lack it. When researchers transferred Akkermansia from responding patients to non-responding patients, the previously non-responding patients began to respond to treatment. One bacterial species made the measurable difference between immune response and immune failure. Pomegranate is one of the most effective dietary sources for increasing Akkermansia. The dose from the studies is 8 ounces of pomegranate juice daily. Make sure it is 100% juice, not a juice cocktail with added sugar. Pure pomegranate juice is tart. If the intensity is too high, dilute it with equal parts water. You can also eat the seeds directly, sprinkled on salads, stirred into yogurt, or eaten by the handful. Within a month of consistent consumption, Akkermansia levels can shift measurably. I put together the protocol I would hand to a patient walking out of my office, because I have spent 32 years watching women work harder than they ever should have had to, against a process they did not know how to name, and because most of the women in that office looked exactly like you. The compound that standardizes the most studied mechanism across all 5 of these foods is a ceremonial green tea extract standardized for EGCG concentration, not generic green tea powder with undefined potency, not a diluted shelf product with no standardized catechin content. The standardized extract with the concentration the clinical studies used. The same compound that reduced inflammatory markers by 30 to 50% in 8 weeks. The same compound that increased natural killer cell activity by 47% in 3 months. Prepared as a daily capsule, taken every morning before breakfast with a full glass of water. I use and recommend this because it is the closest commercially available formula to what the clinical research actually supports for women over 60. And because the women on this channel deserve the same answer my patients get. Now here is the complete daily protocol, built from the research. Morning, 2 cups of green tea with breakfast, brewed at the correct temperature, steeped for 5 minutes, or 1 teaspoon of matcha powder whisked into hot water. Mid-morning, 1 cup of fresh or frozen papaya. Lunch, half a cup of broccoli sprouts on a salad or sandwich, or 1 cup of steamed broccoli, including the stalk. Afternoon, 1 additional cup of green tea. That brings the total to 3 cups, which is the therapeutic dose from the studies. Dinner, purple potato as a regular rotation, baked, roasted, or as a soup, cooked and cooled for resistant starch benefit, then reheated gently. Before bed, 8 ounces of pomegranate juice, diluted with water if needed. The gut bacteria work through the night. 2 women came to me with histories that may sound familiar. Helen, 66, Atlanta, Georgia. Helen's mother had breast cancer at 68. Her older sister was diagnosed at 63. Helen had mammograms every year and felt beneath that discipline that she was simply waiting for a diagnosis that was inevitable. What Helen did not know is that genetics defines risk, not outcome. The internal environment defines outcome. In 8 weeks on this protocol, Helen's inflammatory markers showed the largest reduction her physician had seen in years of routine blood work. The physician asked what had changed. Helen had changed the environment. Donna, 71, Seattle, Washington. Donna had been eating broccoli every week for years. What she had not known was that she was discarding the stalk and using boiling water, which destroyed a significant portion of the sulforaphane before the first bite. The correction was technical, not effortful. Same food, different preparation. In 12 weeks, oxidative stress markers in her routine labs had shifted measurably. Donna did not change what she ate. She changed what she kept. 1 in 3 women will develop cancer during her lifetime. After menopause, when natural killer cell activity declines and chronic inflammation finds less resistance, the internal environment shifts, silently, without announcement. But the environment can be rebuilt, not with dramatic interventions, with 5 foods in specific doses, prepared correctly, consistently. Chronic inflammation creates the environment cancers need. Weakened immunity fails to eliminate what the environment allows to grow. Impaired anti-angiogenic defense allows the blood supply that turns a harmless cluster into a clinical tumor. The 5 foods address all 3 links in that chain, every day, before breakfast, starting now. The biology does not pause while you decide. The research is in the journals. The protocol is specific. The link is in the pinned comment. The only variable left is whether you begin today. I have pinned a comment directly below this video with the exact product, the exact protocol, and the exact timing written out in plain language. It is the first thing you will see when you scroll down. Everything you need is there. Do not wait.